Hey everyone, so I have my first anatomy exam of the semester tomorrow, and I'm trying to really do good on it, so I'm making this video to show you guys how I plan on studying for it, and I'll let you know if any of these habits work after I take my exam tomorrow. So it's about 10 p.m. tonight. I have about 2 hours before going to bed. It's important to get a lot of sleep before an anatomy exam or just about any exam to just be ready. So some of the things I'm going to be studying tonight before jumping straight into it is I'm going to be referencing my anatomy textbook. I'm going to be using my notes from class. Yep. My notes from class. And I'm going to be using this whiteboard to write down certain topics that I read through or that I find in my notes or in the textbook that are just very hard for me to grasp at the moment. So I'm going to be putting that all on here. Another important thing is to take practice tests. If you know any upperclassmen who have taken tests before and your professor is very much known for changing questions on exams, consider asking them to borrow their tests so you can, practice. As long as the professor is not is constantly changing the questions on the exam, so you're at least challenging your mind, because the main point about studying, learning anatomy is you have to be able to apply it in any situation. So if you, it's important to realize your weak points and be able to reason and come up with ways how certain things are all connected, kind of like a story. If you're able to do that, I know a lot of people who take anatomy are trying to succeed in the medical field, me myself as well. So it's important that to not take this class lightly and do as much as you can to understand anatomy. Okay, so the first thing I'm going to start with is I'm going to be starting with my in-class notes. So from the first day, I'm going to be running them through you guys. Okay, so first day of classes, January 9th. First thing, oh yeah, also sometimes the professor will give you a bit of an overview of what's going to be discussed on the exam. So an overview would look something like this, list of things to study. So I'll just read this off for you guys. So the list of things we need to study for this digestive tract exam is an overview of the digestive tract, structures of the upper and lower gastrointestinal tract, and its accessory structures, the general functions, general patterns of tissue layers in the gastrointestinal tract, the 4 layers and the subdivisions of each one, the mesenteries, their names and functions. Then the next pointer is the oral cavity. So what are some of the functions of the oral cavity, the anatomy and specific functions of each structure, the esophagus, the tissue layers and its functions, the stomach, general functions of the stomach, gross anatomy, epithelial cell types and functions, endocrine cell types and functions, gastric problems, regulations of gastric activity, so knowing the 3 phases, stimuli and response for each phase, the small intestine, so the gross anatomy of the small intestine. So there's 3 sections of the small intestine that I need to be able to understand for tomorrow. Epithelial, submucosal cell types and functions, secretion and absorption that is happening in the small intestine, the 2 types of movements happening in the small intestine, such as peristalsis, segmentation. The pancreas, the histology and digestive activity, regulation of activity, the gallbladder and the liver, the anatomy and histology of the gallbladder and liver, the 4 categories of liver functions, summary of secretions into the small intestines, so a list of things that are secreted there in the small intestine, and the large intestines, such as the anatomy and histology, functions and activity. And finally, my professor wants a summary of the absorption of nutrient groups, metabolism, by the liver and transport by the bloodstream. Now, hearing all this information at once in this overview really does feel like a lot at first, but I can tell you right now that if you just point out your weak points, you'll be able to get through it all very easily. So just looking at this list already, I can already point out some of the things I want to be able to understand right now. So I'm going to put them on the whiteboard I have here. So things I don't really understand include layers of the digestive tract. The next one would be the, something I don't really understand, would be the pancreas. Another would be the gallbladder and liver. Secretions in small intestine. Large intestine functions. Liver metabolism. Nutrient groups such as vitamins, transport by bloodstream. Okay, so now I have written out, I'll just show you guys right now. So I've written out the weak points I have right now for this exam that I don't really feel very clear about. So I have to be able to keep these in mind. So now I'm going to be going through the actual my actual notes that I've read this overview of my weak points, and I found my points. So going into my notes now, okay. Some of the things we talked about in class on the first day. Digestion is the process of the body breaking down ingested food into molecules small enough to pass through the intestinal wall / cell wall of the stomach, membranes that line the digestive tract. So each in this book there are some figures that are being talked about and mentioned. It's important to also open them up as you are reading to help you understand what you're talking about in your notes, because at least my anatomy professor would always open up some images in class on the PowerPoint. She doesn't really use slides, she's more on putting pictures up. So the picture that was shown first is figure 24-1. It's major organs and accessory organs of the digestive tract. Okay, some food items never pass the cell wall but are still part of the digestive process. By being defecated / released from the body as waste products because they are only digested to a certain size, they cannot be absorbed. Digestion as a stopping point. Once these small molecules get absorbed, no longer digested, it involves post-absorption and then transporting these molecules crossing the membrane into cells that line the tube, the digestive tract. They will next either help cells that line the tube or move the nutrients from the cells to the bloodstream. So first step in digestion, it has to be able to go down that tube, absorb through the cells and into the interstitial fluid where the vessels are, and into the bloodstream. Digestion begins during ingestion, putting food in your mouth. So there's lymph vessels where lipids enter. What goes into lymph? Nutrients. Nutrients are absorbed to cells that line the tube and out to the bloodstream. Nutrients enter hepatic portal system, so liver, from the digestive tract. The liver will then make many decisions. Metabolism, so onto the metabolic story. So there's anabolizing / catabolizing. So anabolizing is a making bigger molecules from smaller ones, and catabolizing, breaking down complex molecules to smaller molecules, or smaller molecules to smaller molecules. Okay, the structure of the digestive tract. The digestive tract has an oral cavity, so your mouth, which is used for, so these are the major organs. The digestive tract has an oral cavity, your mouth, which is used for gestation, tasting, chewing, or mastication. The esophagus, not much going on here. The stomach is a lot is going on there. Then the small intestine, which is divided into 3 parts, and the large intestine, also known as the colon, which has several parts. The pharynx is at the back of the throat, where food leaves the mouth. There's 3 types of pharynxes, so oral pharynx is important right now. It has stratified squamous epithelium there. It is involved with the mouth, for scraping cells at the back of the throat. So these are the organs I mentioned are the major organs of the tract. Some of the accessory organs of the tract include the teeth, the tongue, salivary glands, the liver, the gallbladder, and the pancreas. Okay, so onto the interior of the digestive tract. So there's distinct layers and tissue layer patterns. So the first one is the mucosa, which has layers covering the epithelial layer, and a thin backing of connective tissue. There's a mucous membrane. It's not serous, cutaneous, or synovial. It's stratified squamous or simple columnar in the digestive tract. Its purpose is to repair abraded thermodamaged surfaces. Simple columnar housing glands to secrete and absorbing from the lumen. Lamina propria for back, which is the back. Oh, these are the parts. These are the accessory parts of the mucosa. I'm still continuing on the mucosa. Lamina propria is a backing of connective tissue where only capillaries enter. The muscularis mucosa is the muscle layer of the mucosa. It is smooth muscle. Its sole purpose is to form ripples. Ripples form when contracted, forming little peaks, mixing absorption, operation, and abrasion. And we also have a swallowing reflex going on. So the epiglottis prevents food from entering, and we have sets of muscles that prevent food from going down the trachea by having the epiglottis flap on top of the glottis. Food behind the liver to the duodenum. So I'm just going to summarize all the layers. So far I've been only talking about one, the mucosa. So all the layers there's 4 of them. So there's the mucosa, the submucosa, the muscularis externa, and the serosa. So MSMS. So the submucosa. The submucosa is the main connective tissue layer. So there's loose connective or connective tissue proper here. It houses most of the blood vessels involved in the digestive process because the epithelium is avascular. The nervous system plexi, or the autonomic nervous system, is innervating there, causing movement going on. The autonomic nervous system, smooth muscles. More onto the nervous system functions of the digestive tract. So it contracts under autonomic nervous control for mixing and churning, peristalsis, causes glands to secrete upon signal, controls blood flow to the digestive tract. Onto the parasympathetic division, it doesn't do blood vessels, so it is all under blood vessels are all under sympathetic control. Under sympathetic influence, the smooth muscles at the core will dilate and blood vessels to non-essential places will constrict. This is done to send blood to the core. So sympathetic is more constriction of blood vessels sending that blood to the core. So the core includes the brain, the heart, the respiratory tract, and the muscles that are using them. So the third layer of the digestive tract is the muscularis layer, or the muscular layer, or the muscularis externa. Not to be confused with muscularis mucosa, which is in the mucosa layer. So that the first layer is the mucosa. So muscularis layer has a circular layer of muscle inside and a longitudinal layer of muscle outside. There's a serosa, so the serosa is serous membrane. So there's a bit. The next figure we're going to be talking about is more on the mesenteries. So the serosa is serous membrane. It's double-sided connective and epithelial tissue sides. The serosa secretes serous fluid into the interior, and center is secreted, serous fluid. So only when the body needs blood, it uses the sympathetic division, which allows the muscles to move, or in fight or flight situations, blood is sent to the core. The digestive system is a non-essential periphery, so when no blood is needed, sympathetic system will relax. Blood vessels to the digestive, reproductive, and urinary systems, dilating the vessels. So parasympathetic is not involved with blood vessels. It's more on the digestive stimulation processes. Going on into the mesentery. So there's about five layers of peritoneum. So there's a figure in the book of the different types of mesenteries. Okay, so there's a lesser omentum, there's a mesocolon. Mesocolon is more on the colon, or the large intestine. The greater omentum is more on the covering the stomach. I'm looking at a figure right now of the digestive tract, and and there's also a lesser omentum in the back. The small intestine is held together by mesentery proper, holding it to the back, and everything is the entire peritoneum covering all the digestive organs is parietal peritoneum. There's a falciform ligament holding the liver to the diaphragm also. So the mesentery is back-to-back layers of peritoneal membrane, bridging the space between the dorsal body wall. So there's bridges of double-sided peritoneum. So the serous membrane is So some of the functions. So falciform ligament connects the diaphragm to the liver. The mesentery functions. So the mesentery keeps the digestive organs in place. An example would be doing a handstand. Your organs wouldn't fall out because your mesentery proper is keeping your organs in place. It acts as a bridge. Mesentery acts as a bridge through which blood vessels and nerves can travel to the organs. They act as avenues for blood and nerves to each organ. The next one is the lesser omentum, which connects the inner curve of the stomach, connecting to the liver. The greater omentum on the outer curve of the stomach, connecting it to the large intestine. It's a flap peritoneum. Mesentery proper has hepatic portal vessels, which are yellow-dyed vessels. The mesocolon are the folds for the large intestine's version of a mesentery. It connects the large intestine to the dorsal body wall. So the 5 names important to know are falciform ligament. the greater omentum, lesser omentum, mesentery proper, and the mesocolon. Onto the oral cavity. So some functions of the oral cavity include mastication, so chewing to begin digestion, salivating, secretion of the 3 sets of salivary glands, so parotid, sublingual, submandibular glands, salivary glands. They also have lysozymes, which is also in saliva that lies bacteria. There's another function is, so some of the functions we discussed so far: mastication, chewing, salivation, secretion of enzymes in the saliva and the tongue. So some of those enzymes also used to reduce bacteria. So lysozymes in the saliva that are lysine bacteria. You also have tonsils in the back of the throat, which have crypts that capture bacteria, so that lymph system has an idea of what is if there's a foreign type of bacteria entering. Swallowing. So a bolus is a ball of food being swallowed, must be able to make a safe passage down the oropharynx and into the esophagus without slipping into the glottis and into the trachea. You have tongue, cheek, and lips to position food. We have hard and soft palates. Okay. So the next time we had class, we looked at a different figure. The figure we looked at was one on the oral cavity. So it shows the cheek, the palates, the gingiva, the vestibules, the tonsils in the back, the oropharynx, important things to look at. So knowing the understanding a figure of an oral cavity. So some of the functions of the oral cavity in part 1 is secretion of enzymes. So the first enzyme that's important is salivary amylase. Salivary amylase is used for beginning the digestion of carbohydrates, mainly starch. An example would probably be rice. It's secreted from the tongue to keep the tongue watery or polar as opposed to non-polar. It keeps the tongue. Salivary amylase keeps the tongue from getting coated with. The second enzyme is lingual lipase. So lingual lipase digests lipids, so it keeps the tongue from getting coated with lipids, in order to allow the tongue to do what it's supposed to do, which is mainly tasting. Lingual lipase starts the digestion of lipids. Okay, another function of the oral cavity is it acts as the first defense against bacteria or ingested food. So we have lysozymes. Lysozymes are secreted to help decrease bacteria. It lyses the walls of bacteria. Okay. It's embedded in the epithelial surface. So lysozymes is an enzyme in bacteria, and the other two enzymes are salivary amylase and lingual lipase. So the tonsils have folds in the wall called crypts that have lymphoid nodules embedded in their surfaces. Lymphoid nodules contain lots of lymphocytes that identify what is coming into to gear up the immune defense against non-self antigens. So the bolus is swallowed at the crossover between the gloss and the epiglottis, then down to into the esophagus. That's the food you ingested going down your esophagus. Swallowing is a combination of nerve innervation, so both autonomic and somatic nervous system are innervating both skeletal and smooth muscles. Also, coordination of events separating the swallowing of food from the inhalation of air. Onto teeth. Humans have Humans have a primary set of teeth, which is your baby teeth, about 20. Adults have the second set of teeth, or the permanent teeth, which is about 32. We have 2 full sets of teeth. The second set comes at age 6. So the teeth sit in the mandible and maxilla bones in sockets called alveoli. So there's 3 regions for teeth. There's the crown, the part that the tooth sits above your gums, the neck, the narrow part of the tooth partially covered by the gum, and the root. The teeth also have dentin, which is under the enamel, also like enamel. If it is acellular, it is not a biological cellular substance. We also have a pulp cavity where blood capillaries and nerve endings sit. There's a periodontal ligament cushioning between tooth and bone. There's cementum that cements dentin into the alveoli of the bone by a couple of structures, cementum and periodontal ligament. A ligament is made of dense fibrous connective tissue. The gingiva are gums of the living mucosa. So there's a space between the gingiva and the tooth where bacteria like to hide and secrete a sticky matrix around themselves called plaque. Plaque is secretions and decaying food. So if plaque is allowed to calcify, it will become a harder version of plaque that is difficult to scrape. It is called tartar. The tongue has skeletal muscle tissue going in 2 different directions. So saliva is a combination of both serous and mucus secretions. So there's a serous acini. So serous cells are a working clump of cells, little circles of purple stained nuclei, which secrete serous fluid. Serous and mucus acini end up in ducts that carry the fluid from parotid gland to an opening in your oral cavity. It depends if you're eating highly acidic food from your saliva, you will become more basic. So if there's a low pH in your food, very acidic food, then your saliva will be very high in pH, very basic. Sodium, potassium, and bicarbonate and antibodies also exist in saliva. We have an immune response, so tonsils recognize antigens, antibodies being produced and secreted in saliva, and the enzymes lysozyme and amylase. So the esophagus. I'm just going to run through some of this and just spit out the important parts for you guys. There's not too much stuff that I really think I don't understand here. I guess something important would just be enzymes produced in the oral cavity. That's a bit of a vague topic. I'm putting this right now on my whiteboard. those enzymes are very important in terms of digestive processes. So there's about 2, excluding lysozymes. So lingual lipase for lipids and salivary amylase for carbohydrates. I'm putting carbs. So this is how it's looking right now in terms of things that I'm going to understand. Okay. Other than that, the major digestive organs are pretty self-explanatory. Still working on it. Mucosal layer. So the mucosal layers include the epithelial, lamina propria, and smooth muscle. Okay, something interesting. So different from a lot of things is in terms of tissue, the esophagus is a very interesting one. So the esophagus has stratified squamous, epithelium, the esophagus also has no serosa. Instead, it has adventitia. Then there's also an acid reflux. If you have a gastroesophageal muscle, if your gastroesophageal muscle is not constricting properly like it should, you get heartburn or reflux of gastric acid from your stomach, causing heartburn. Okay, on to the stomach. So some things that I found really interesting about the stomach is its ability to sampling the chyme. Very interesting fact about the stomach is its protein breakdown. That's been a very hard topic for me to wrap my head around. So the stomach is focused on the breakdown of proteins. So high protein meals means the food is going to stay longer in your stomach because peptides hasn't broken those peptides that are making up those proteins. Another interesting part of the stomach is that it produces an intrinsic factor which allows you to absorb vitamin B12 from dairy products and vegetables. So intrinsic factor is something I don't really understand much about, but would be important to know. So vitamin B12 is important for erythropoiesis, or blood cell production. So once food goes into the pylorus, the name changes from a bolus, the food name changes from a bolus to chyme because it's about to enter the duodenum, or the small intestinary. So there's two types of sphincters. There's an esophageal sphincter, which is on top of the stomach, connecting the stomach and the esophagus. And there's a pyloric sphincter connecting the stomach and the duodenum, or the pylorus. Something I don't really understand too much is the muscular layers of the stomach. So something interesting here is that the stomach has 3. So there's an oblique, circular, and longitudinal. Oblique, circular, and longitudinal. So something interesting here is oblique muscles on the stomach. So oblique muscle is the innermost layer, which is used for churning and mixing. Churning and mixing. That's something I don't really understand which muscle does that, but it's apparently the oblique muscle layer of the stomach. Stomach has dips called gastric pits. And deep into those gastric pits are gastric glands. And gastric glands, so gastric pits mainly have mucus cells for mucus secretion, and gastric glands have more secretions. So lamina propria has both lymph and blood capillaries. So in the stomach, the muscularis mucosa is responsible for maintaining the sides of the rugae. Rugae are folds in stomach directing food downward. So when I say gastric, it means stomach. Enteric, if I say enteric, it means I'm talking about the intestine. So gastric gland cells, there's a couple of these, but they're very important. So some of the gastric gland cells include the parietal cells, so some of the things they do is secrete HCl, lowering the pH, secrete intrinsic factor, secrete hydrogen and chloride ions. That become HCl in the lumen. Something important that happens is from the parietal cells is the alkaline tide. So that's sodium bicarbonate being formed in that process. So moving on to another figure. We're on to the stomach now. We see a figure of the secretion of hydrochloric acid. So hydrogen and chloride ions combine to form HCl once it enters the lumen of the stomach gland or the gastric gland. Alkaline tide happens in the interstitial fluid, where chlorine enters the parietal cell, but bicarbonate is taken out of the cell, which increases the pH of the interstitial fluids. So some of the story that's happening here is onto the story of the alkaline tide. So cells release carbon dioxide into the bloodstream, combines with H2O in the blood plasma to form carbonic acid, then to hydrogen and bicarbonate. Hydrogen ions go into red blood cells and binds with hemoglobin. Bicarbonate ions are kicked out of the red blood cell in exchange for chloride ions, so that was the chloride shift. Bicarbonate is safer to have in your blood plasma than is carbonic acid. Hydrogen ions get sent into the lumen of the stomach instead of into the lumen of hemoglobin. So what do strong acids and a lot of heat do to proteins? So strong acids denature proteins, breaking the bonds that hold protein in a globular form and kills bacteria ingested. Also activates secretion by G cells. So G cells secrete a proenzyme or precursor enzyme, inactive enzyme, pepsinogen. So pepsinogen is the inactive form, and it's activated when HCl to form pepsin, protein digesting enzyme. So pepsin works on proteins denatured by HCl. It starts the breakdown of primary bonds between groups of amino acids. Proteins to amino acids, or AA. So in summary, gastric pits are mucus secreted, or secrete mucus. They have a very high pH of 8 or 7.5 pits. Parietal cell secretions include intrinsic factor in hydrochloric acid, which messes up the bacteria, has hydrogen and chlorine ions secreted in combined messes of bacteria and starts to break down the proteins. Denatures them, activates pepsinogen and active pepsin. Chief cells secrete pepsinogen. Gastric pits. The end, nice blend of active enzymes, active acids, bacteria that are dying, and proteins that are being broken apart. A few bacteria that can survive, that they run into trouble in the duodenum, where the pH is suddenly a pH of 9 and 9.5. Any bacteria that survives a pH of 2 will not survive 9. Stomach problems. In infants, living on a diet completely of milk, there's only one protein to worry about. Infant chief cells also produce chief cells, besides secreting pepsinogen, do a couple of other things in special cases. Infant chief cells also produce a special kind of milk digesting enzyme called renin. So renin denatures milk proteins. Infants also secrete gastric lipase in addition to lingual lipase. Designed to take care of milk fat denature. Depending on what species you determine how much gastric lipase depends on how much lipids in the mother's milk. The pylorus of the stomach has more secretions. There's pits and glands that have secretions from G cells called gastrin. Gastrin is an endocrine hormone. Hormones from the stomach are secreted directly into the bloodstream. So I'll just show you guys a bit of how my progress is looking on the board right now. So these are some of the things I need to be able to understand for tomorrow, or recall for the exam. Things that I have a hard time understanding, or feel like are very difficult concepts to grasp or remember. I'm trying to re-jog my memory in the morning before the exam, basically, after getting a good night's sleep. So a bit of the process. going to stop this video right here, but one quick note is an important thing to study when you're studying anatomy is study your quizzes that are given in class, because a lot of times your anatomy professor will base your exams off of your quiz questions. So I'm just going to conclude the first half of this video here. Thank you guys for studying with me, and I hope you all do well on your anatomy exams. Good luck.